Tumor.
Malignant.
Second male test?
What do we mean when we say cancer cells can become immortalized?
You don't know.
Because they make their
But
own blood supply and things like that. They get their own nutrients
No,
from.
that doesn't immortalize them.
Telomerase?
Telomerase, they turn telomerase back on. Did that? No. Something else. I think I'm getting all of
So
them.
telomerase inhibits or enhances?
Which one?
Telomerase.
Telomerase turns back on. It turns the enzyme, when you're young and growing, especially in embryology and everything else, your cells go adding really fast, right? So you start whittling down, you would start whittling down those telomeres too quickly. Telomerase rebuilds the telomeres.
So it adds in the sequence.
It adds to the length, yeah. So that, the idea is that you don't, you're never gonna damage the actual chromosome after replication, right? Because the telomeres get replaced. So like end caps on there to keep it protected. Now, they're trying some treatments to give telomerase to people.
For fountain of youth?
Yeah. That'd be a good idea or bad idea?
Bad.
Yeah, pretty much live forever, don't they?
We're overpopulating.
Yeah, that's the ultimate problem. I think we're already overpopulated. Can you imagine everybody living to be 150? But what's another potential issue with it?
Cancer.
That's what it said. It's pretty much an anti-cancer mechanism, right? So now, you may have had a couple of tumors in there that didn't turn telomerase on, eventually would have died, and now you've just done what?
Turn them on.
You just immortalize those tumor cells somehow. So yeah, I'm not sold on the whole telomerase thing just yet. Okay. Anaplasia is used to describe the loss of cell differentiation in cancer tissues.
What side is that?
It's the back.
Oh.
Am I the biggest? Oh, sorry. Sorry. I know, it's the biggest. Genetic instability, what's an aneuploidy from 1406?
The number of chromosomes,
What about it?
when there's too many or too little.
Right, it's a missed number of chromosomes for whatever reason, aneuploidy. How many are we supposed to have?
24?
How many?
46?
24? Wow. How many total?
46. Wait, or
How many?
40 something. 46?
Going once, going 46, going once, going
46, 46.
Oh, 46. We are diploid, we have two sets of 23, right? There you go. Insertions, deletions, point mutations, and short repetitive repeats, okay? These are all genetic instability issues, mutations that can cause problems. Growth factor independence. We talked about this before we looked at it. Lab prep guys out there. Growth factor independent. Most of your cells, we talked about that with androgen, remember? Most of your cells need growth signals to keep growing. That's part of it. It's the whole part of the regulation. Cells should only grow when they're getting the appropriate signals to grow. Okay? If you get growth independence, growth factor independence, you no longer need those signals to keep growing. And then we talked about cell density dependent inhibition or contact inhibition or loss of their. Your cells, when they get too crowded, they get signals to quit dividing. Otherwise, your organs would just do what?
Keep dividing.
Keep growing theoretically, right? So that's all part of this contact inhibition or dependent inhibition.
So they have to, I don't understand that, so they have to be touched.
Well,
They stop
that's
growing.
what's weird about our cells. That's what's so, they've got both. Right? So we'll look at this one first. And the good example, like we said, is the tissue culture flask. If I put some tissue in here, it's going to start growing until it covers the entire bottom of the flask. Normal cells will quit growing at that point because of contact inhibition. They know they're too crowded, the density dependent inhibition, whichever one you call it, they know they're supposed to stop. Right? Cancer cells lose that. So they start growing and they'll actually start growing on top of each other.
And the normal cell was,
Normal cells
Die
won't,
or
in fact,
stop.
normal cells will actually start to die.
so that's just like a signal that says stop growing.
You get all this contact everywhere else, you get, start sending out signals, we're too full, we need to quit growing. Cell cohesiveness and adhesion, that's the flip side. Your cell tissues like to be in contact with other cells as well. They have that cell-cell signaling going on, so they like to be in contact. That's the hard part about growing what they call primary cells. Is you don't get enough in there, they don't like to grow with each other, so we have issues. So it's both, right? You've got to have some cells for ours to grow, but when they get too many, they stop. Cancer loses both of those. So I can theoretically take one cancer cell, put it in a tissue flask with the right media, and it'll do what?
Grow.
It'll grow. Anchorage dependence is another one. You also, your cells, all these different mechanisms, want to be anchored down to the connective tissue and stuff below it. They like, that's all part of being an organ, right, or a tissue. You've got to be nice and together. Do your cells break off and suddenly float through your body? If they did, what would happen to them? They actually end up dying, they signals to die. Cancer cells lose that anchorage dependence as well.
So do they die because they don't have nutrition or because
They start losing the signaling. You have all these signaling things on the outside surface, and like the gap junctions and all that cell signaling, when they start losing that, they're like, whoa, something's wrong. And your default mechanism if something goes wrong is apoptosis. We think something's wrong, we do what? Right.
Kill it.
And again, this all goes back to cell-cell communication. Faulty, altered gap junctions, altering signaling pathways, increasing of receptors so they require much less stimulation to grow, loss of contact inhibition receptors, all this different signaling, cell-cell communication is changed. And then we talked about this part, the lifespan, they become immortalized, or they can become immortalized. They turn telomerase back on. And this is the key you're looking for too, we're talking about right here. This is the golden goose or whatever you want to talk about. Antigen expression. Most cancers they're finding out now make some sort of antigen other cells don't make, okay? Not all of them, not that I know of, but a lot of them do. We just can't narrow them down. The point is if you can narrow them down, you can do what?
Prevention.
Two different things. You can detection, early detection, you can get soluble markers for it, blood, so like. Some lab last year claimed they could detect almost every cancer from a blood sample. That was, yay, that's the last I heard of it. So that means probably what? Somebody was sitting somewhere or something, and the lab in Israel said they cured cancer. Yeah, I haven't seen that one either.
So when a cancer starts from the whatever cell it started from,
The stem cell, the cancer stem cell?
is that what that is? They know where it started, it was like in the liver or the.
They can still kind of tell, they can still tell, they don't undifferentiate completely, so you can still tell where the primary tumor started, yeah. So if I have a lung cancer cell that metastasized to the liver, the second, oh, sorry, over here. Secondary one, right? You can still tell it was originally what?
Oh, lung cell.
Lung cancer. So you can still, because the biopsies can still identify the primary tissue.
I was a little confused when you went over the anchorage dependence because cancer can travel around your
That's why
body.
so it loses its loss of anchorage dependence.
But
Yeah.
some cancers can stay in that one.
They can, but they don't have to.
Okay.
They can still survive if they break off and metastasize.
Oh, I thought you were saying that they die. And,
No, no. So most
Oh.
cells have anchorage dependence.
Okay.
They can't survive if they lose. Cancer cells lose that. They don't. Doesn't matter anymore.
Oh.
So they can.
I was reading that wrong.
A
I
lot
get
of these
it.
all have to do with metastasis, don't they? When it really boils down to how they can survive when they're not supposed to.
And that's when it's stage 4, right?
Well, it depends on how far it's metastasized. Usually metastatic cancer stage 4. Because the other ones, technically it's metastasis, but usually you think about moving around the body. And then you also get these production of enzymes, hormones, cytokines, all these different weird things they start producing they're not supposed to be producing. Because they lose that differentiation, right? They don't know what they're doing.
Mm-hmm.
So cancers can produce all this?
Some of them do. Some actually produce some toxic enzymes start damaging the actually surrounding tissues and themselves sometimes, so.
So they have their own network, own blood, everything?
They can, if they do angiogenesis, they can a lot of times, yeah. Now, not all of them do this, this isn't the hallmark, all of them, a lot of them do, not
Yeah.
all. And it depends on the tumor. If you have, okay, what's Cushing syndrome?
That's the hormone one, isn't it? It's a hormone imbalance, isn't that
Yeah,
one?
so usually caused by pituitary, a pituitary tumor is relatively benign, but it starts producing too much what?
Oh,
Starts producing too much,
Is it
the
androgen?
ultimate, no, cortisol. So you start
cortisol.
getting too much cortisol production.
That's the one where the eyes bug out, right?
Yeah, but they get the moon face, and they get a little.
The
fat
pump?
Predisposition, it's, my dog has it.
What?
Dog?
Are you kidding?
No.
Oh my God.
Let's take a little pill every day.
I
Oh,
thought you said your daughter. I was like, why is he laughing?
No, no.
So if they're in like the breast cancer, it could produce
Yeah.
estrogen?
Yeah.
Yeah, so look at things like that.
and then that's how it even causes cancer in like the uterus. It because it doesn't necessarily travel, but the hormones, right, or something like
Well, you
that.
could get a tumor, that'd be a secondary tumor, that
That's
would be
a
a
good- yeah.
secondary primary.
That's
tumor,
what I
actually,
meant.
would be a secondary tumor. But prostate cancer, sometimes can start producing too much DHT actually, which is gonna be self-stimulating to grow and everything. All right, and cytoskeleton changes. This is where they become invasive and metastatic. That means they can start invading neighboring tissues and that causes damage. Or they can become metastatic and start moving through. So the cytoskeleton is just what?
The filaments?
It's shell, so it gives it a
Shell.
shape and allows for movement and then acts like that rail system. So it can start changing in there.
But that's the myosin, right, the actin?
No, that's in the muscles, mostly active, it's mostly gonna be,
So it's just cytoskeleton of the cell?
of the cell itself, yeah, not down the side. All right, so those are the big hallmarks of cancer right there in terms of cell morphology in physiology. Okay?
So this is the cell morphology.
Yeah.
So that's just different ways the cancer survives?
It's just, yeah, different mechanisms that are characteristic of cancer cells. So a lot of them are morphological, a lot of them are just, they lose these different dependencies, like they're supposed to keep tissues in check, right? Your tissue's not supposed to break off, go somewhere else in the body and keep growing, right? That's not supposed to happen. All right. So direct invasion and extension, we talked about this. So everybody talking about the enzymes they can secrete stuff, so they can secrete enzymes, and then they insert these enzymes, they're gonna start breaking apart cell-cell adhesion molecules. Desmosomes, tight junctions, things like that, they help hold together. And then they start inserting these crab-like extensions. They start just sort of squeezing their way through the tissue and invading other tissues. Benign tumors don't do that, right? They may grow, but what are they gonna do to that neighboring tissue?
Stop.
They may squish it, but they're not gonna invade into it. They're not gonna break out of that primary tissue and start moving into it.
So is that would that be similar to like virus, how they spread and they inject themselves, or?
Not so much here, because this is more like, I'm trying to think of something more like an example. It'd be more like an infection itself spreading, but not the individual bacteria themselves. It's kind of moving out and starting to spread out now. So more like I think I have a picture I put in here. It has, now you have no clear borders. So this is a problem when it starts doing this, because if you want to do surgery, what are they trying to get?
The cancer, so they can't see it all.
But what do the surgeons look for when they're looking, that they send, every time they're cutting out, they're gonna send, you guys will be there, they'll send a slide down to the pathologist to look for what?
Abnormal cells.
No, they already know they're abnormal cells, what are they looking for?
Like the capsule or?
The cap, yeah, some of these don't have a real big capsule, but they're gonna look for the abnormal, the borders, what they call them, or the margins, right?
Oh.
They're gonna start looking, so they're gonna keep cutting until every pathology slide comes up with clear margins and clear borders.
Oh.
They have the capsule and everything else.
I just assumed they could see that with CAT scans and.
No, because now you're looking at, so when this little migration, you're talking about very few cells, not big chunks of the tumor, very few cells actually moving in this, initially at the end. So if it's kind of like a crab-like projection here, and it's moving into this tissue like this, I mean, the very end down here, you're just gonna have a few cells down here, right? You're not gonna have this big tumor.
Oh, I see what you
This thing may
mean.
flatten out even more.
And that's what they have a hard time
So
seeing.
now they wanna make sure that when they surgically remove it, they don't do something like that. Now you've removed it. what do you do
They're trying to get
after?
the root out.
Trying to get everything out, you wanna get everything out, that's why they try and use what now? Afterwards, after surgery, what used to be the common
Radiation?
chemo and radiation to try and get anything they missed. We'll talk about that at the end of the class. Now what do? Now what they've actually flipped this around a little bit. The idea before was you cut out the tumor, you hope you get it all, and you try and blast what you hope you missed. Blast what you hope you missed? No, not hope you missed. Blast, hope you hit what you missed, right? Now, they're trying to hit them, because these are the fastest growing parts. They're trying to hit them with chemo. They're trying to hit them with chemo and radiation before surgery, and they could try to shrink
Oh,
the tumors now.
that's right.
So they can have a better chance at finding those clear margins and those clear borders. So now they're doing pre-surgical radiation.
Wasn't the only way one.
Huh?
Wasn't the only way one.
All right,
No.
seeding of cancer cells in the body cavity. That's another problem here. Once these things start to move through, if it goes through your intestine, if it's colon cancer, and it breaks through the intestinal wall, what's gonna happen?
It spreads into your.
Won't it allow bile to leak out if it's going through your
No, because this is
intestine?
gonna be kind of a solid thing, it's gonna kind of move through it, you don't have too much.
But if it breaks your intestinal wall,
It becomes more.
like you can die faster.
Well, you could, but usually it's not going to, usually it's gonna actually seed cancer cells. But you can start dumping cancer cells into there, and now it's got more room to what? Move around and stuff
Oh, okay.
inside there. Called seeding, not metastasis, so the interperitoneal cavity. Ovarian cancer, then you get what?
Fluid buildup.
Fluid buildup, because what's one of the hallmarks of ovarian cancer we really see? What are the symptoms of ovarian cancer? Almost nothing.
Yeah.
What are the symptoms that a lot of women have later on?
Like you get full
They
easily?
feel full, you feel full because you're getting this buildup of fluid in your interperitoneal cavity. So it's starting to fill up, so you'll eat a small meal and you'll feel real full, so that's one of the early ones.
So is that why
I feel like that,
some
I don't have
cancers are,
ovaries, so I don't think.
is that why some cancers are easier to remove than others? Because of the borders and where
Yeah,
they're
oh yeah,
located?
yeah, if you got, some cancers are much easier to, like prostate cancer, as long as it hasn't metastasized, prostate cancer is not too bad, you just go in and do what?
Take it out.
Take the whole sucker out, right?
The whole prostate.
Yeah, well, we have radical prostatectomy, yeah. The problem with that is, what runs right through the middle of your prostate.
We all look clever.
Why are men so reluctant to have radical prostatectomies? Not even partial one.
Well, doesn't it what's sexual function have to do with that?
It's just, central function is just to dump fluids into the semen to decrease the rate of pH.
Urethra,
Yeah, yeah.
the urethra, that's.
Yeah, it goes through, that's not too bad, you can miss that, that's not too bad, that was pretty good, you can get around that one. The nerve that runs right through it though,
That's for
that
you.
has problems,
It's hard on.
that triggers erections, yeah, so a lot of men, in the past, it was almost about 95% were impotent afterwards.
That's
So-
why it's not
Wow.
sexual function.
Well, I mean,
In a nice way.
die, take a die, I mean, I can. Because if you're dead, it's not happening either, so I don't see what the.
Time to think about this, please.
Metastatic spread throughout the blood or the lymph, this is the bad part. Remember, most people die from what? Primary tumor or
Metastatic.
secondary tumor? And then the sentinel lymph nodes, we talked about that, they're gonna be the lymph nodes that are gonna be right around where that tumor is. Okay? That's what they're gonna look for first. And we talked about it in AP, we didn't talk about it here. What's one of the problems with breast cancer when they do surgery on these sentinel lymph nodes?
Oh, the fluid buildup after.
Well, where though?
In their arm, because they're taking out
the
the pit.
lymphedema in the arms and everything else, because you get rid of these lymph nodes, so it just basically blocks off the return lymph flow, and it just pulls back in the arms. All right. Uh-oh. Guess I better, yeah. And this is just showing this whole process right here. So what's going on here? Here's your primary tumor, right? It's malignant. Contact with the basement membrane, so it's gonna start secreting enzymes and stuff so it can do what?
Get
it
inside.
can move through and get out of there. So passage through the extracellular matrix, going through here, moving through all this, because the extracellular matrix is not really solid, right? You can move through it. So it starts moving through, now it's gonna get where?
Oh, into blood.
So this is gonna be invasiveness, and now it's gonna get what? Into the lymphatic system or the blood system, it's gonna spread, and then you get venous what?
They're too tiny, I can't read it.
It gets, where does lymph end up eventually? That's the easiest way to go.
It is.
In your venous system, right? And once it gets in your venous system, it goes right into the heart and then it goes everywhere. So anchorage to vessel wall, you can't read these very well, so don't worry about it. All they're just trying to show here is what? So down here, I'll just walk you through it. You've got this, tumor gets in here, lymphocyte, platelets, get the segregation, but now you get what? Secondary tumor that's moving out of the bloodstream, and now you've got a metastatic tumor. But this is in where? That extracellular matrix. Is there a lot of blood supply in your extracellular matrix just floating around?
No.
No, so you're gonna start doing angiogenesis now. So now what's it gonna do?
Grow itself.
It's gonna start growing its own blood vessels, so now it gets its own blood supply and its own oxygen. And if this happens to be a tissue down here, it can actually do what? If you got a big enough tumor and it's really trying to grow, because remember, it's going through glycolysis, not cellular respiration, so it's using a ton of what?
ATP.
Ton of glucose to make that ATP, because it can't make much of it. So it's constantly going through glycolysis and burning up glucose. So they can actually and that's one thing that happens, they start starving the tissues around it, because they can start sucking all the blood off.
Mm.
Okay.
Red blood cells are, I was seeing that, they're, don't proliferate, right?
Red blood cells?
They're selectively different, or terminally differential.
Yeah,
So
yeah.
I was a little confused on that, so.
On the red blood cells?
Uh-huh.
Okay, so they
Because they
mature,
come from the stem cells.
Right.
So when proliferation really means regenerate themselves,
proliferation just means to make lots of copies of yourself, to grow a copy.
and then red blood cells don't.
They
Like,
don't.
They die off and they,
You make more, but where do you make them from?
the bone marrow.
The bone marrow. So they're made in the bone marrow, and the last step before they go out and do their job is actually get rid of the nucleus.
So they don't go through mitosis.
Not after that, no, they're done. That's why they only live, what, 120 days? Yeah, 120 days, because they can't, they've got just enough machinery in there to keep them alive that long.
So how do they carry cancer?
You don't get this.
Oh, so it's not, it's just in the blood, like.
Yeah, this is just
In the
going
lymph.
blood, yeah, yeah.
Oh.
Yeah, no, I've never heard, now,
So when the cancer's in the
Never say
blood,
never.
like the leukemia and all that, that's the white. blood
that's all, the white blood cells, yeah, or the stem cells in the bone marrow. I think it's all the white stem cells too, for the most part. Now, I'm not, I think there may be one that's a red, let me check. So as long as, before they actually eject their nucleus and everything else, when they're back, when they're growing, you become red blood cells, you could maybe have a cancer starting in one of those lineages, so I'll have to look on that. I think there is one. but
Yeah,
it's
because
pretty rare.
that's why it was a little confused, 'cause it says kind of that they don't proliferate,
Mm.
and I thought also.
So their only job is to do what?
Carry
out.
The
oxygen.
last thing they do, they get made, they cram them full of hemoglobin, take out the nucleus, and say, okay, go carry blood for a while. I'm gonna carry oxygen for a while.
So when they proliferate, are they in a certain cell, what phase are they in the cell cycle? Do they stay in like the gap, any of the gap phases?
Once they're mature red blood cells, they're in G0. They're out.
G0,
Yeah,
so
so they
they're
don't even
like
have the
they're-
mechanism to do anything
Okay.
anymore. So the closest thing you would be G0, yeah. Because they don't have the information to copy.
So similar to neurons, but it's just that we make more.
Even worse, because these things you couldn't even coax these guys to reproduce. Now they're thinking that there's some ways you can actually coax neurons to reproduce. These guys, there's no DNA, so could they reproduce even if they
No.
wanted to? Yeah,
No.
So they're really not even a cell anymore, are they? They're just bags of hemoglobin.
I never thought of that. That's true.
All right, so primary site of tumor, and this is what you guys will see, because some of these things become almost diagnostic. Breast cancer, well, axillary regional lymph nodes, contralateral breast via lymphatic, so it goes to the other side, right?
Uh-huh.
Lung, pleural, liver, bone, and brain. These two likes to go through a lot, likes to go to the bone. Metastatic breast cancer of the bone is not a pleasant situation to be in, because what happens when it gets inside the bone marrow?
Well, it gets the stem cells.
Now, you can't turn things to cancer, but what happens inside of there?
It's
They
taking all
start
the
growing
nutrients.
and growing and growing, and your bones like to move. So it's actually, metastatic bone cancer is incredibly painful.
So if you have like lung cancer and you never smoked, it's not usually a primary tumor?
No,
Is
it
that what I'm
It's
saying?
a primary tumor, yeah.
So why isn't it on there?
But hold on, we'll get to the next time.
Okay,
See,
sorry.
that's like-
Just kidding.
That's it. Colon, regional lymph nodes, liver, likes to go to the liver, lung. Also likes to go to the stomach right here, and the urinary bladder because it's nice and close, right? But this is the one that likes to go to the peritoneum. It likes to infect other tissues, other spaces. Kidney, lung, liver, bone, adrenal gland, ovarian, peritoneum again because of where it is, right? Regional lymph nodes, lung, and liver. Everything likes to go to where?
Lung.
The
what's
liver.
that?
That's what we're saying,
But everyone-
because all your blood goes through the liver eventually.
Oh, that's true.
Pancreas, this is just bad. Liver, stomach, by direct spread, it's not even metastatic there, it just moves into the stomach. Colon, lungs, peritoneum, prostate, bone, that's the problem, prostate likes to go to the bone too. Regional lymph nodes go down, now which one were you talking about? Oh, lung. I don't know why they don't have lung up here.
Looks like it'd be important.
Somebody doesn't like lung cancer, I guess. Where's
Yeah.
the lung like to go? Lung likes to go to the liver, everything likes to go to the liver. They also don't have, the skin cancers up there either.
Yeah.
Kind of weird. Where do skin cancers like to go? Where does
Lymph?
melanoma like to metastasize to?
Oh,
Some bone a little bit, let's go to the brain. That's a big problem, it likes to get in the brain.
Why?
I don't know.
They just like
Maybe
it?
I don't know the way they can change. Well, that's pretty, yeah, fine, right. Anyhow, yes, anytime, any of those cancers you're talking about, where the tissue they develop in, that's a primary cancer, the primary tumor. Secondary is when they branch off and go somewhere else.
So when they move somewhere else, the DNA is still the same from the.
Tissue's still the same too. So if I have, well, I can't have that, so I'm not gonna
It's
do it.
just growing in a different location.
It's just metastasized.
So no cancer starts in the liver.
Yes, you do have primary liver cancer. It's called HTC, HCC, though. T, why did I get T in there? HCC, hepatocellular carcinoma. It's really not that common. Your liver's pretty good at normally dealing with stuff like that because it regenerates. What are the things that can cause primary liver cancer?
Oh, scarring, inflammation.
Okay, but what leads to that? Cirrhosis. Cirrhosis can cause you that cough inflammation. So either alcoholic cirrhosis, cirrhosis from non-alcoholic fatty liver disease, or
Hepatitis, right, for the inflammation.
and there's another one, I can't think of whatever it is. Anyhow, hepatitis, yeah, what you said, yeah, I'm sorry, yeah, hepatitis, viral, it's chronic inflammation can cause that. Those are about the only three. Now, there are a few chemicals out there that can do it. Carbon tetrachloride will do it, aflatoxin. What's aflatoxin? Where do you find it?
Was that the Roundup we were talking about?
No, that's glyphosate.
Oh.
Glyphosate. That's lymphoma.
Oh,
Where do you find aflatoxin, everybody? You know?
give us a hint.
You eat it.
Eat it. Is it like that wax they put on fruits and vegetables? No? Okay,
It's got a lot of peanut butter and stuff.
oh,
It's a fungus that grows in the soil. You get too much aflatoxin, it's big time liver cancer causer, so. I would be very careful about making my own peanut butter. I'm also, I don't know.
those are like corn and stuff like that.
You could get some, you cook some of those, pretty heat, really well. All right. So factors affecting tumor growth, the number of cells that are actively dividing or moving through the cell cycle like you were talking about. Not all these cancer tumors are always gonna be doing that. Some will go to G0, they'll stop. So the more you have in the cell cycle, the faster the growth is gonna be, right? The duration of the cell cycle. If you're a benign tumor, a lot of times they'll do what? After a while, what happens to benign tumors?
They'll stop?
They'll stop growing, they'll just quit growing.
So it's not that they do it faster, like, than a regular body, they're just more.
Sometimes it's faster too. We also replicate faster a lot of times too. It just depends on how fast the tumor, you hear about fast growing tumors and slow growing tumors, it just depends on what it is.
Cause they don't die.
Right.
Because of the.
So you get in there. So if you have someone that become, growth factor is definitely dependent, someone else will start growth factors. They don't need signals, so they constantly have those turned on signals to replicate. Okay, the number of cells that are being lost with the number of new cells being produced, again, your immune system actually cleans up a lot of these tumor cells. It knows they're not supposed to be there. Some of them will actually shut it down after a while. And growth fraction, which is the ratio of dividing cells to resting cells in tissue mass. Right? Basically, what's going to affect growth, tumor size, is how many, how fast, and how many are done. Okay, and that's all that is. And doubling time is the time, which is the length of time it takes for the total mass of cells in a tumor to double. Now, this is where you're going to get kind of scary. Okay, and the growth fraction increases, doubling time decreases.
Well, yeah, because that,
Huh?
yeah, that makes sense because,
Right, so growth fraction with the ratio of dividing cells, resting cells. I thought I had it, so right here, you usually cannot even detect a tumor until it's gone through 30 doubling times. That's when it becomes physically apparent, when you can detect it. And that's what? How many cells?
trillion?
100,000, right? I mean, a million. A million. Like 35 doubling times has gone up to what? So it's big enough to see at 30.
Oh, it's three different numbers. It's 1,000,035
Right.
and, okay, 1 billion,
Yeah,
is that?
it should be,
Oh.
So 5 more doubling times, it goes from what?
1
A
million
million to
billion?
a billion cells. So that's how rapidly they can do this, if they grow fast enough. And that's the problem with tumors. Why is this problematic, this growth curve? Or tumor growth, why would it be a problem?
Well, it's
For
best to
research.
catch it as early as possible so that you, before it doubles and starts moving around and stuff.
So all tumors are 100% curable if you what?
Catch on time.
You catch them early enough. So the problem is you can't see it like this. 5 more doubling times, you've done what? I forgot what I need to look up. The fatality rate goes up exponentially almost when you get to this.
Just by 5?
So that means 5 more doubling times. It goes from just barely being detectable.
But how would you detect it? And then-
This is where you can actually see it like on imaging and things like that. and everything else.
Well, what would make you image it?
Well, that's the problem. That's the bigger
There's
problem.
nothing else
Okay,
in the
this is
blood.
saying, this is the earliest you can detect it. This is when it starts to really becoming a big problem already.
But is there any other indications in the blood?
That, the tumor markers, what I was talking about, that's the key they're trying to find. So because ideally, way before you even get to this, those tumors should be secreting those tumor markers in your blood.
How
Well,
long would
do-
it
do
take to
they-
get to that stage?
Depends on the cell type. I'll have to look up, I'll get some examples of them. Because some of them are really slow growing, other ones are really fast.
So when you're talking about.
But
Do the cancer patients, like, do they get any other signs or symptoms before a tumor?
not usually in this one, not usually at this stage. I mean, think about it, this is barely detectable. And it's gonna depend on where it is, too. The problem with ovarian cancer, pancreatic cancer, what is the problem with those? By the time you get signs and symptoms, they're usually what?
You're stage
Oh,
4.
It's usually too late, yeah, that's the biggest problem.
So when you're talking about cancer types,
Mhm.
it just depends where it originates from. That's why it's so hard to cure, because there's so many.
Well, there's also 100 different mutations in there. This cancer has a mutation here that's causing it to be cancer. This one has a mutation here, this one's got P53 mutated.
Is treatment all the same?
Okay, that's a good question. Is treatment all the same?
No.
No, so what are the treatment options we have now?
Of course, chemo, radiation,
So what's
surgery.
chemo?
Chemo is, it's pumped into your system.
It goes all around.
It goes all around, but what does chemo do? What's its mode of
kills
action?
you.
Kills the nucleus?
No, kills you.
Kill-
Enough.
It kills
Enough.
the cells?
So that the
Cancer?
cell loses its supply of like nutrients and oxygen.
No, that's not, because that'd be bad for you if you got that full.
Yeah, that's why people die of or
But they
come
can't,
close
well,
to
I
it.
can cure any cancer with chemo, I just have to pump enough in it to kill you.
Yeah.
To kill cancer.
Yeah.
Isn't it just like suppressing all the cells that are growing in your body?
Their chemotherapy, the one traditional chemotherapy you guys think of are cytotoxic drugs. So they kill rapidly dividing cells. They go after cells that are rapidly dividing.
Mm-hmm.
That's why hair falls out, you get gastrointestinal problems, you
Oh.
get
Oh, 'cause those
anemia.
are the cells that are.
All those are rapidly dividing cells, that's where all the side effects come from. So you get immunocompromised because your white cells get killed off, right? Your red blood cells start to die off, you get anemic, hair falls out, you start having diarrhea and stomach problems because the mucosal intestinal lining starts dying off. And that's the problem. How much can you pump in there to get before you kill the cancer cell? So those are good. Those are all just cytotoxic, they all work. Some mode of action is to kill rapidly dividing cells, but indiscriminately, right? What are the other ones? Radiation is basically the same thing.
Target, but it's
just-
more of a target.
It is because you can target more, so it's not as systemic, so in that instance, it's a little bit better, but you can only get cancer cells that are relatively close to the surface.
Yeah.
If I have a pancreatic tumor, you wanna blast it with radiation?
No.
Because you gotta go through all those other tissues to get to it, right? Now, the new drugs are called targeted therapies. They're either immunotherapies, where they're training the immune system to re-recognize or to recognize the tumors and launch an immune response. PD-1 inhibitors, PD-1 ligand inhibitors, okay? CAR T cells. Or you have these targeted therapies which target particular pathways in particular tumors, and that's what I'm talking, you get particular. I say particular enough, okay? These target, if you have a P53 mutation, they can go after that. If you have a KRAS mutation, actually, I can't go after that yet, but that works.
Is that the CRISPR stuff you were talking about?
Hmm?
The CRISPR stuff you were always
Well,
saying?
that's some of what they're trying to do, but these are just, they're trying to, so you've got a mutation in your ch6 pathway. Okay? This is one of the growth pathways, particularly like lymphomas, leukemias, and stuff like that. So this signaling pathway gets turned on, and that's just telling those tumors to keep what?
Growing.
Growing, right? They've got some drugs now that just target this particular pathway. So now is it gonna get any other cells? It's not gonna get as many. Get some immune cells, but those are the new targeted
Is it the
therapies.
JAK inhibitor?
Yeah. Mhm. So they're trying to get these things, there's also,
'Cause I see those for like autoimmune.
they do that some too, because it's usually, the ch6 pathway is big in the immune system. So that's why I said the lymphomas and leukemia, some of them. They've also got kinase inhibitors. Kinases are two phosphorylate pathways, so they're targeting in particular kinase. So those are the new big ones they're trying to do. The idea is they're hoping in 10 years from now, they can take your tumor out, they can look at its genetics, see where the mutations are, and then prescribe a drug that's only gonna go after that particular pathway that needs to fix it. That's what they're hoping for. I think the immunotherapies will be, I think they're gonna win that out, but
Those are bad.
don't do me, every stock I buy tanks, so don't buy it. Don't listen to me yet. All right. We're only in 7 minutes. All right, let's go through this right here. Open this up right stop it. This kind of goes through what we're talking about for targeted therapy. So proto-oncogenes, what's an oncogene? Cancer gene, what do
Oncogenes,
oncogenes do? Why do they get this notorious name? It's kind of unfair, they're kind of like names this because it's bad. When you're growing, they're genes that are designed for growth, right? And they're supposed to get turned off. So proto-oncogenes are growth factor, receptors, signaling molecules that promote growth and division. C-mix, one of them. Okay? Diet can have a lot to do with this. Because what will diet do? What have they found out about diet now and cancer?
Isn't it that cancer likes sugar or something? That's what I.
Loves sugar because cancer needs what? Lots of glucose, so it loves sugar. So what are the new treatment options they're looking at? Doesn't require any drugs at all.
The diet, ketogenic
The
diet.
ketogenic diet, they think they may see some response in tumors to the ketogenic diet because the tumors can't get enough energy. They can't